So George also works on organoids in the Geschwin lab at UCLA, um, and he has done a lot of work on KMT2C, which he's going to talk about. And um, Dr. Kleefstra on the first day of the conference talked about KMT2C and how they have been doing work at understanding the clinical presentation of KMT2C versus EHMT1. And KMT2C has been called Kleefstra syndrome too, but their conclusion was that when they did the clinical evaluation was that KMT2C really deserves to be like its own thing and not be like a subset of Kleefstra syndrome. And so now they're discussing really should it get its own name, like its own syndrome, because it really is deserving of being a separate syndrome and not seen as like another type of KS. However, if you saw Jackie Harris's presentation, she was talking about writers and syndromes of writers, and KMT2C is also a writer, just like EHMT1 is a writer. And so in that sense, they are definitely related conditions. And so we thought it'd be really interesting to have George talk about the work that he's done on KMT2C. And I think your intent, George, is also to start looking at EHMT1 and doing some compare and contrast between those two at the cellular level using these models. So I'll let you take it from there. And thanks so much for coming.
All right. Well, thank you so much, Eric. Thank you, Jeff, for the invitation to share the work that I've been doing. Thanks to those of you who have stuck around all the way to the end. My name's George Chen. I'm a postdoc in the lab of Dan Geschwin here at UCLA. So over the past 5 years, I've been leading a project in our lab that has been establishing stem cell lines from KMT2C mutant patients and characterizing their development using cortical organoid culture. So it was very apt that I follow Angel's excellent talk and describing this whole process and how the organoids are particularly useful. So since this is our lab's first time in sharing all our work with you, I really want to quickly go over the scope of our research, how we became interested in KMT2C. I'll talk about the characterization work I've done so far with the KMT2C lines that we've established and where we plan on taking this project, and bringing it together with a larger study with EHMT1 and as well as the other writer syndromes, as Jackie had mentioned yesterday. So this conference has really kind of run the gamut and really brought together a lot of researchers that have very shared interests, and I think we can support each other in helping this rare disease community. Our lab, the Geschwin lab, has broadly been interested in understanding the genetics of neurodevelopmental disorders with a focus on autism spectrum disorders. And so, as many of you all well know, many forms of genetic variation contribute to the presentation of ASD. And over the last 2 decades or so, hundreds of risk genes have been identified, largely through identifying de novo mutations. And this is probably not news to this community in particular, but these monogenic forms of ASD are largely either known to or predicted to have their mutational impact through haploinsufficiency, where you have one copy of the gene that is mutated and the remaining single copy of the wild type gene is insufficient to maintain normal function. And so we had hypothesized in our very early work that we could use CRISPR activation to correct the expression level by targeting the enhancers of these particular genes. So enhancers are regions of DNA that do not directly drive the expression of a particular gene, but seem to regulate that whether or not that expression increases or decreases. And the underlying goal here is to take that haploinsufficiency, where you have a insufficient amount of gene expression, and restore it to wild type levels. And so for CRISPR activation, I selected a dCas9, which is a dead Cas9, meaning that it doesn't cut the DNA, and it's linked to acetyltransferase P300 as our activator. And we chose this particular activator out of the many CRISPR activation constructs, as this one was predicted to be a medium strength activator of gene expression. And it's normally used to target enhancers, and we use that to target a guide RNA to increase the expression of the remaining wild type copy. And so we focused on enhancers because haploinsufficient genes require this delicate balance in gene expression level. And so from this study in 2019, looking at 1,000 different haploinsufficient genes over the whole genome, we can see that the haploinsufficient genes have a much tighter regulation of gene expression. So the cell-to-cell variability is significantly lower than the rest of the genome as a whole. And additionally, the group found that while underexpression in the case of haploinsufficiency leads to decreased fitness, overexpression leads to cell toxicity. And so this suggests that the existence of haploinsufficiency may be conserved within evolution as a mechanism for stabilizing gene dosage. And so just to kind of again, this is setting the stage for how we got into KMT2C. We started with this workflow for identifying mutant phenotypes and then targeting them with CRISPR activation. First, by finding a gene of interest, and then going in two arms. The first to identify mutant phenotypes, and then the second to target enhancers, and then with the hope of establishing a phenotypic rescue at the end of it. So I'll first talk about the characterization of the organoids first. Um, as you heard from Angels just a moment ago, the cortical organoids are stem cell-derived 3D cell cultures. They've been demonstrated to recapitulate key developmental processes of the prefrontal cortex. Here I'm showing you some immunofluorescence and clarity images from a 200-day-old wild-type cortical organoid, just to demonstrate to you that the different cell types all exist within the organoid, and so they become a very useful way of looking at neurodevelopment, especially in those early time points when we can't have access to precious brain tissue. And so we first targeted two really well-known ASD risk genes, CHD8 and SCN2A. For CHD8, of course, we start by confirming that we have established lines that have a reduction in CHD8 expression. So the wild type lines are in blue and the mutant lines are in green here. CHD8 haploinsufficiency is associated with macrocephaly, which we observe in the organoid cultures here. So again, in blue are the wild type lines, in green the mutant, and the mutants at matching time points are significantly larger than that of the wild type line. And we showed that this increase is due to an overproliferation of intermediate progenitor cells. And so here I'm showing you the size of the EOMs positive cell population, and this is using EOMs as a marker of intermediate progenitor cells. So these are 2D images of the organoids stained for signal intensity of this particular cell type marker, and you can see that in the CHD8 mutant lines, it is much more pronounced than that of the wild type line. For SCN2A, as it's a sodium channel subunit, we focused on neuronal phenotypes. We, of course, show that the lines have a diminished amount of SCN2A. We see that because it's a neural marker, this marker SAPB2 of upper layer neurons shows a significant decrease in reduction, particularly at these kind of late maturation stages. And there's a diminished firing pattern in these mutant excitatory neuron cultures. And so through these experiments, we confirmed that we have identified a target gene, they have reduced expression of that particular gene, and there are mutant phenotypes that are observed in organoid cultures that are consistent with that is seen clinically with patients that are affected by these mutations. And then just to move down to the second arm of this study, we then look to identify target enhancers, and this takes advantage of a lot of work that other people in our lab have done by building maps of enhancers using human fetal brain tissue, which we then identified 4 target enhancers for CHD8, 3 target enhancers for SCN2A, and after a bunch of filtering to try to identify the best case candidates for guide RNAs, we were able to come down to a set of 2 guide RNAs that seem to work for CHD8 and 1 for SCN2A. And so what we find with this CRISPR activation treatment is that in the organoids, we were able to significantly increase CHD8 expression, which we show here. Here's the control targeted line, and then with the two CHD8 guides, we're able to see a significant increase in the gene expression. We see a significant decrease in organoid size, and we actually see a correlation between the level of CHD8 expression with the decrease in size. And so that's really satisfying to see that we are able to reverse that phenotype. And as I had mentioned a few slides ago, this is due to an overproliferation of intermediate progenitors, which we measured by the marker EOMs, and we see a significant reduction with treatment. Down to near wild type levels. Here's the wild type in the bottom left corner here. And we see an increase in expression of an immature neuron marker. Here I'm using DCX. In SCN2A, we see an increase in SCN2A expression, as well as increases in neuron marker genes, TUJ1 and MAP2. And most importantly, we see a rescued firing ability. So here I'm showing you what the wild type looks like. This is what the mutant looks like. And with the rescue, we see a near complete rescue of the firing activity. And so taken together, we find that enhancer-targeted CRISPR activation is able to rescue both gene expression and mutant phenotypes. And so this is kind of a proof of concept work. It really got us excited to explore how broadly this approach could be applied. And at this time, we had a patient that was willing to provide their cells for stem cell reprogramming. And after doing whole genome sequencing, we identified that that patient had a mutation in KMT2C. And so this was our entry point. And I took this opportunity to study this gene in particular, and it has since expanded into its own project. And so my plan to study KMT2C haploinsufficiency, as well as Kleefstra syndrome and other Reiter syndromes, builds off of the work that I just presented. But given that CHD8 and SCN2A are very well characterized, whereas KMT2C and EHMT1 are much less, although this community has done so much work already in demonstrating finding out more about what these two genes do. I'm particularly interested in comparing the shared and distinct consequences of mutations in KMT2C and EHMT1, and then apply the CRISPR-A based system that I just described to rescue gene expression and see if this is a valid approach, valid therapeutic approach for these two disorders. So over the past, I would say 5 years, I've established a cohort of KMT2C mutant patient stem cell lines, thanks to collaborations with the clinicians here at UCLA, as well as our undiagnosed disease network. And we've paired them with neurotypical control lines. And then the EHMT1 line that I have here is the same line that Angels just described. And we are also just starting to integrate it into the study. So I won't have, I'll have one slide of data to show you on that one line, but the rest of it will be on the KMT2C lines that I have, that I've spent most of my time characterizing. First, of course, we want to validate that we have reduction in KMT2C expression, and we do in all 4 of our patient lines, both at the RNA level as well as in the protein level. And one phenotype that has been observed in studies of EHMT1, and this is a study that other people at this conference have referenced, was that EHMT1 mutations lead to a dysregulation of neuron maturation, in which the premature maturation observed leading to an increase in the number of neurons, but a decrease in the neurite length. I find a similar result using an NGN2 driven neuron system. We find increases in neuron markers DCX and MAP2 relative to that of a control line. So here in dark blue. And so we established cortical organoids from all of these patient lines, and we observed that the mutant organoids are smaller than the control lines at matching time points, mirroring the microcephaly phenotype that is observed in some patients. And paired with the observation that the rate of neuron maturation is increased in these mutant lines, it suggests that there's a premature cell cycle exit, which depletes progenitors early in development. But we see already at day 50 days, I show significance. Day 25 also has significance, so I don't show that data here. And here's my one EHMT1 slide. These organoids are also significantly smaller than control. So Angels, here's the data that you were referencing. You just needed to collect. You're welcome to it. But I replicate see what you see as well in terms of the decrease in gene expression of EHMT1. Um, and we see a decrease in size there as well. So we've collected organoids at multiple time points, starting at 25 days post differentiation, all the way up to 200 days. And we've characterized them primarily using bulk RNA sequencing, but we're just starting to use spatial RNA sequencing as well. And I'll describe a little bit of the work there. And as you can see, the bulk RNA sequencing has all of the lines that I have, 3 controls and 4 KMT mutants, whereas the spatial one was a pilot study, so it only had 1 control and 2 of the mutants. I'll start by talking about the bulk RNA seq data here. Here I'm showing principal component analysis of 90 total RNA samples. This is an analysis that allows us to quickly see if there is a variable that contributes significantly to the variation between samples. And so on the left-hand side, I'm showing you the PCA plot of all of the samples where they are colored by age. And we can see that there is the largest degree of separation because of age, with the youngest organoids over here on the left-hand side, with the most mature organoids over on the upper right-hand side right here, the 200-day-old organoids, and there's kind of a split on the x-axis. When we look at mutational states, whether they are the control line or the KMT2C mutant line, we do see separation, although that takes second fiddle to that of the age the age division. So once you break these samples down into all of one time point, you see that the distinction between mutant and wild type there. Taking these all together and looking at their differentially expressed genes between the control lines and the KMT2C mutants, I use DisGenNet, which is a database of gene disease associations that is sourced from the literature as well as curated repositories. And I show a collective enrichment in terms associated with neurodevelopmental disorders as a whole. You can see at the very top, but also terms associated with Kleefstra syndrome phenotypes. And it suggests that the organoids are recapitulating certain key aspects of in vivo development. Additionally, we see a marginal overlap of upregulated differentially expressed genes with autism risk genes. So here I am using the Simons Foundation Autism Research Initiative list of high confidence autism risk genes here, and then our significantly upregulated genes and the significantly downregulated genes from the bulk RNA data as a whole. And we see a marginal but significant amount of genes that are represented in the overlap here. And this is consistent with a mouse model of KMT2C mutation, which also showed that the upregulated ASD genes were present in the adult mutant forebrain. So then, because the control versus KMT2C mutation seemed to be a phenotype that was best characterized on a single time point level, I divided the data set into the individual time points and then compared the control lines to the mutant lines, and then looked at the gene ontology of their across these individual time points. And what we find is there is this inconsistent change in neuron maturation rate, with day 25, 150, and 200 showing gene ontology terms that are more mature than that of the control line, that there's an enrichment for synapse organization in ion transport, in synaptic organization again here, whereas the day 50 and day 100 time points showed a decrease in maturation compared to the control. Those same terms showing up here in the control line. So downregulated here meaning relative to that of the control. So we therefore decided to compare our organoids to the human cortex to understand how these organoids compare developmentally across these time points. To do so, we used a method called rank-rank hypergeometric overlap, and we compared our data to the Allen Institute's BrainSpan data set, which is taken samples of human cortex across development from prenatal stages all the way to adulthood. And so in this analysis, just to orient you as to how this works, you'll see these color plots or heat maps and a high degree of overlap in the upregulated or downregulated genes leads to an increase in this intensity of a red either up here in this quadrant or down here. So overlap in upregulated in the top right, overlap in downregulated in the bottom left. We've previously used this approach to show how organoid development compares to in vivo data. So again, using that BrainSpan data, I'll show you the set of stages are all prenatal samples and then postnatal. So there's a transition right here in the middle. And our organoids, which we grew out to 600 days, we were able to show that there is a certain amount of maturation that occurs. And there's hints of increased maturation past day 200 that resemble postnatal, but not very strongly. And this relates to what Angel was just saying, that at these time points, as we get further along in development, other cells start moving in, and vasculature is required to allow the cells to mature more. And so there is a max limit, there appears to be, for organoid maturation, but we can still model quite a bit, especially in these early stages where samples from individuals are rare, if not extraordinarily rare. And so we first find that our control lines largely resemble brainspan stage 4 at around 50 days, with one of our control lines kind of shifting down to stage 5 at day 100. So basically, again, we look some of the samples we didn't have all of the time points. This one we only had two of the time points. Um, but this is consistent with what we have seen in other control organoids, that they basically roughly model this kind of late prenatal period of time. By comparison, two of the patient lines appear to have transcriptomes that are most similar to brainspan stage 7 at day 100 onwards, which is interesting because this is that prenatal to postnatal transition. And these two lines, although they're not very strongly tied to these later stages, they're definitely the most significantly tied compared to all other time points. But we had also had two lines that showed a different pattern where it appears there's a global delay in maturation in which these lines are much closer to the stage 4 that we see in the control lines as well. And so, although all of this is very preliminary data, and there's a lot to be done still in this bulk RNA analysis. Taken together, what we show is that the KMT2C mutant organoids exhibit premature neuronal differentiation. As a whole, they recapitulate key features of neurodevelopmental disorders, but we do see patient-specific differences in rates of differentiation that we'll still need to understand. And Niall referenced this at the very start of today, that there are patient considerations that need to be taken into account. His model beautifully illustrates the ability to use iPSCs and patient recordings from the same individual to really tailor therapies down to the single patient. And our study here, at least on a broad scale, suggests that there may be value as well in looking at those kind of at the single patient level. Of course, different cell types are affected to a greater or lesser extent by various neurodevelopmental risk genes, as others have alluded to at this conference. And so we've begun to use spatial transcriptomics to explore how cell type specific changes in KMT2C mutant organoids are established. And so, as a means of validation, we compare our spatial data to that of immunofluorescence of the same organoid, and we can see that our cell type marker expression largely agrees with the cluster annotation. So here I have four we call fields of view or FOVs, in which each point is a cell that has been sequenced. This is the exact same organoid, albeit a serial section of that organoid, stained for the marker genes MAP2 for neurons, PAC6 for radioglia, GFAP for astrocytes, and then DAPI to stain the nuclei. And so we can see a good degree of overlap between particularly the excitatory neurons here in mustard on our spatial single cell, with that of the MAP2 expression, which we see over in the immunofluorescence. Um, what we aim to do next, of course, is to perform community network analysis to identify how these different organoids are organized, and if there are changes related to the mutation there. So just kind of as a kind of broad overview, we first initially processed all of the single cells together. So I'm here showing you what are termed UMAP plots. Each point here is a different cell. On the left, they've been grouped into clusters, which we were able to annotate into the different cell types, as you had just seen in the previous slide. On the right, it's colored by which section of tissue they've come from. I got a total of 47, I believe, FOVs. And so this helps communicate that there are certain FOVs which are overrepresented in particular cell types. And that's largely because spatially, you take either the edge of the organoid, you'll have more of a certain cell type versus the core of the organoid, you'll have more of a different cell type. So this is not unexpected. We then perform what's called pseudotime analysis to try to line up all the cells that we've sequenced along a developmental trajectory as a means of showing rates of differentiation. And so this approach shows the least differentiated cells on the right, the cells that are labeled for radioglia, and then they branch outwards towards on the left, and then they branch outwards towards the right-hand side into the different differentiated cell states. And so this was really satisfying to see, as it suggests that we are able to capture cells from across the entire range of differentiation in the single cell data that we have. And when we perform differential gene expression on this data, we're able to look at particular at specific cell types and how their gene expression is changed in the controls relative to the KMT2C mutants. And here we see that the biggest change in the upregulated genes, these are upregulated in the control relative to the mutants, are in the astroglial population. In particular, they seem to target HIF1, which is hypoxia, and then pathways of neurodegeneration. That one's a little bit too broad, so we'll need to drill down further to understand what that encompasses. Uh, but by comparison, when you look at the downregulated genes, this which downregulated in control, meaning relatively upregulated in the mutant lines, this seems to largely agree with what we have shown in our bulk data, the increases in synapses in the mutant organoids. They are largely in affecting these radioglia populations as well as the excitatory neuron populations and the neuron population and then the XN, so these are the excitatory neuron populations. And so this shows that there are cell type specific effects of KMT2C mutations and that we can start to tease apart. We can also start to look at the effects on cell neighborhoods. So not only single cells are interesting, but single cells don't exist in a vacuum, particularly in an organoid where they are so tightly packed together. There's lots of cell-to-cell communication. And so people have started to develop algorithms that try to group these cells together into neighborhoods in which these here we're now coloring this algorithm referred to them as niches where they have grouped together by similar composition. And by this looking at these similarly composed regions of organoids, we might be able to understand how signaling, how cell-to-cell communication is different in these different regions, and that may also provide more insights into the mechanisms behind the mutation leading to certain disease phenotypes. And so again, early days, lots of data. Unfortunately, still a one-man show in terms of trying to do all of the analysis. So I have so much work to do. But both global and cell type specific changes we've observed in the differentiating organoids. And I need a few more spatial fields of view to really determine changes in organization. And so we're working on that right now in terms of repeating the spatial experiment with more organoids, and including the EHMT1 organoids, because we now have those in culture as well. Looking into the future, one of the challenges with organoid culture is the high degree of heterogeneity between samples, which is in part due to how the organoids are cultured hundreds together in a dish. To address this, I've been developing a semi-automated organoid culture protocol, which uses robotic systems to maintain organoid cultures. I found this method results in much less variation. We're able to monitor organoid development more efficiently. And so my aim is to kind of build out this platform, and in particular the automated imaging part of this platform, so that we can trace development at a cellular resolution. And once this system is operating at scale, the idea is that this could be then adapted to be used for drug discovery and screening. But just to show the variation in size with our current protocol versus the protocol that is run on this automated platform is significantly smaller. And then just to look at go back to the CRISPR activation part of my talk at the very beginning, I've had some talented students work with me over the past 5 years, and I've set them the task of designing guide RNAs to target KMT2C, and we've been able to significantly increase KMT2C by targeting a promoter as well as two different enhancers. So we've begun to test these constructs in organoids and in 2D neuron cultures. And so there's a lot more to be done. Inspired by yesterday's talks from Jackie and Anna, especially, there's an opportunity to look at how these different syndromes share and have divergent developmental trajectories using organoids. Also be able to understand the impact of these genes on other comorbidities, such as the GI issues that were discussed yesterday and today by caregivers and by the patient families. My goal is that the platforms that I'm trying to build here will not only enable a better understanding of the neurodevelopmental impacts, but also serve as a screening tool which can better serve these rare diseases, particularly KMT2C, EHMT1, and KMT2D. And so, just to close out, I want to very quickly thank my PI, Dan Geschwin. He's given me a lot of support to let this project grow and grow and bring on as many patients as I have been able to have access to. I've had a stellar team of students and technicians that have worked with me on various aspects of my projects. And the various funding agencies who have been responsible for funding my work. And just to close out, I am hopefully starting my lab very soon. I'll be moving to Children's Hospital LA. I'm really excited to be working with Shafali, who Kira had mentioned earlier. I look forward to learning from all of you in the years to come, and hopefully have the opportunity to come back and share my progress on these projects as well. So thank you everyone for your time.
Oh, super cool. I love how all the lines seem to converge in this world, right? That you'll be working with Shafali, so that's great. Congratulations on moving to your own lab. So yeah, I had a few questions myself, and I thought it was really interesting KMT2C how you were really almost getting these like two distinct populations based on the patient variant, and it
Mhm.
looks like there almost are two classes of patients, at least again from what the organoid model can tell us. And I was wondering, I don't know as much about KMT2C. There's been a lot of work on EHMT1 of dialing in what exact variants are doing, and if you have a deletion or a stop
Mhm.
versus an expressed, pathogenic mutation, and then all of that affects, and what do each of those specific changes to EHMT1 really do? And long story short, there are some interesting edge cases, but it still looks like the story is pretty much holding that EHMT1 it's always lost. Like almost always, it's some kind of loss of function. The impact of that loss can be variable, but as far as we know, we don't have any gain of function in EHMT1 that is then causing a totally different situation, right? Which are some syndromes have that where they have both a gain and a loss. And I don't know if KMT2C, if what is known at this point, and if it's possible you have a more complex tapestry with KMT2C, and that might be what you're seeing in your organoid. So that's what I wanted to. That was my question at the end was kind of where does that land? Do you think?
Yeah, it is an interesting question that uh, other people have really kind of highlighted that this kind of loss of function seems to have core known phenotypes that we can reliably characterize, but then there are these edge cases that are particularly interesting and I don't say challenge the conclusion, but put a little bit of color into the conclusion to make it a little bit more complicated. My data set, I have only one patient that has a complex structural variant. Everyone else has a stop game variant, but the majority, I believe, two of these are very close to each other in terms of where they fall on the gene.
Yeah.
The this actually they're all fairly close to each other, but the complex structural variant, of course, is a little bit different, and it does behave differently,
Mm.
at least in terms of neuron maturation. The phenotype is not as strong. The organoid size, the phenotype is also not as strong, despite the fact that on the base level, the KMT2C expression is at the same level as the rest of the patients in the study. So I think there's something there that I'm hoping to tease apart a little bit better with the different bulk approaches.
Yeah. And then putting on our translational hat, which was what Gael was telling us to do earlier in the day. I think the CRISPR-A stuff is really neat. And have you guys thought about what does that look like in terms of being converted to a therapeutic and delivery? Because that's always the challenge with these larger entities.
Definitely. And so this is one where there is a lot of exciting work. Actually, there's a paper that was, I believe, just published last week in Nature, where they did a promoter targeted CRISPR-A to SCN2A on mice. And so they've been able to show that they're able to reverse phenotypes. There needs to be a little bit of work done on the delivery side. And actually, I'm working with that exact lab to to start exploring these differences in these new ways of delivering the system. So, like the
Yeah.
advantage of the CRISPR-A system is that it is an edit, so we can avoid a lot of those concerns about using CRISPR in humans because there's concerns of off-target effects that it'll edit something that it shouldn't.
Yeah.
Here, it's literally just being used as a guide, and so what I think is kind of elegant about the system is that it can go into a individual, hopefully, and just live there and only be useful when it is needed, and then won't otherwise cause any deleterious effects. We're still in the process of kind of identifying what that therapeutic window would look like and how durable that signal is. In my hands, we're able to maintain a rescue of phenotypes in the organoids, at least, for about 2 to 3 months. Um, but I know there are some liabilities of the system that I've used in vitro that would not work in the in vivos model.
Mm.
And I don't believe the animal studies have been carried out long enough yet to see if there is a drop off in their effectiveness. So that's kind of where we're at next stages. I'm independently trying to get the CHD8 and SCN2A working in mice as well, so that we can see if our this enhancer approach works in mice. Um, but the delivery aspect is the delivery and durability aspects are the kind of the next challenges for this approach.
And can you comment a little on the enhancer versus promoter? Because you guys have been trying to do both, and what has actually turned out to be the more formidable way to get a gene turned up?
Right. So a promoter sits at the very beginning of every gene, and
Yep.
basically it's the on switch. And so
Yeah.
you target that, you are pretty much guaranteed if you hit it properly, it will turn on the gene. And the enhancer has additional regulation attached to it. Be it through chromatin modifiers, be it through other means that limits its effectiveness, limits its ability to turn on a gene to developmental time as well as to specific cell types. And so we really feel like this is a great opportunity, again, with the idea that if we just put it in an individual, it will turn on when it needs to be, and it will turn off when it's not needed, to target the enhancers versus the promoter. And so that seemed to be like the more elegant solution, whereas the promoter you would lead to overexpression.
Mm.
That being
Yeah.
said, the study that was recently published, as well as other groups that looked at the promoters, have not shown an overexpression phenotype. And so
Mm.
there seems to be some way that we're not quite sure what that way is that is not preventing that overexpression from
Huh.
happening. And so that hasn't been a concern. Uh, I think it will still be a concern as we move from, cell to animal to human, because it should, by all theoretical means, lead to overexpression. So we'll need to understand why it's not.
Yeah. Yeah. And also maybe their expression isn't net overall, but it's not timely. Maybe it's expressing potentially the wrong timing for that particular cell at this particular moment
Yeah, and so
in time.
one of the things that we've really focused on when talked about our maps is we took human fetal brain tissue and identified these enhancers by comparing that to mouse fetal tissue as well as macaque fetal brain tissue, so that we really enriched for enhancers that are only present in the human fetal brain. And the idea there being, again, specificity to the particular time point of development where it would be important. And that's where we hope to go with KMT2C and EHMT1. Again, one of the other challenges is identifying that therapeutic window. These are developmental disorders. Is this something that will need to be introduced in utero, or is this something where people can get benefit postnatally? That those are all kind of questions that we're just starting to explore, and
Yeah.
hopefully our animal studies will start to kind of address those questions.
Great. So I don't see any other questions in the chat. We brought Angels back up to answer your question since there was kind of a conversation there. So I don't know if Angels, you want to answer George's question?
Yeah, sure. Well, first, congratulations on the talk and on the new position. That's
Thanks.
amazing. That's great. Yeah, no, I would love to. I think it will be really nice to do assembloids for the Cliff-Strauss syndrome, especially the forebrain assembloids to look at the inhibitory migration formation and how that affects. Yeah, I think that'll be really nice. We can talk to the iDefine if they are interested in funding this project also. And I have a question for you if there is time. I don't know this.
There's time. Yeah.
Yeah.
We are still on time here, amazingly. Yes.
Yeah.
Yeah.
Great job. Great job. This, I wanted to ask you about the specificity that you were mentioning about these enhancers. Have you seen any like cell type specific differences? Because enhancers sometimes is specific, right? For cell types. That's one question. And the other question I have about the enhancers is that if you know of any SNPs that have been described or variants in the enhancer that is also linked to disease.
Yeah. To answer the first question, we haven't looked at the cell type specificity, cell type specific upregulation of the genes. I can only say from kind of the crude cell sorting tests that we've done afterwards that at least the excitatory neurons are affected by our targeted approach, and as well as the intermediate progenitors in the CHD8 model. We're still looking at SNPs. That's kind of the next stage that has kind of been handed off to a collaborator to start looking at kind of disease-related SNPs and whether or not we can better identify what to target. So, one of the challenges with identifying enhancers is essentially manual curation and manual testing to make sure that they all work. And so we are trying to make that more efficient using AI models to try to take what is known and integrate that so that we don't have to manually test everything that we find.
And I love your data about the EHMT1 size organoid. That was like this was like compared to which lines did you compare in that experiment?
It's the 1.8 and the 1.9 lines. So I think it's the same
It's
one.
the parent, it's the parent control, parental control, and the and the child.
Okay.
Yeah, okay.
Yeah. Okay.
Yeah.
Interesting. Okay.
And so the EHMT1 premature stop lines from the patient, those were slightly smaller when you grew them, George? Yeah, okay. So they're a little bit smaller. Okay. Yeah.
Yeah,
Right.
it's interesting because we use different protocols, so it will be really interesting to compare between different protocols,
Yeah.
the phenotypes. Yeah.